Hi, my name is Hans Rabi. I'm the Chief Operating Officer here at the Institute for In Vitro Sciences. I and my colleagues are very excited to give overview and presentations on a variety of in vitro test methods for industry, product development, for product stewardship, and regulatory classification labeling purposes. My presentation here today will focus on an introduction to in vitro test methods, an overview of the types of test methodologies utilized from simple 2D cell systems through more complex three-dimensional tissue reconstructions and ex vivo tissues. I will provide an overview of some of the applications and limitations for those test methods. And this will give us a backdrop then for the subsequent presentations from my colleagues. First, Dr. Amelia Costin will give a presentation on skin and skin irritation and corrosion test methods, as well as some thoughts on the future. Next, Ms. Alison Hilberer will give an overview of in vitro test methods used for predicting potential phototoxicants in products and chemistries. Third, Dr. Tinashe Ruwana will give a presentation on a variety of current and emerging test methods for predicting contact allergens, so those that cause allergic contact dermatitis. And lastly, Mr. Nathan Wilt will give an overview of the various eye test methods and some strategies for utilizing those tools and methodologies. So first, I'd like to give an overview of who the Institute for In Vitro Sciences is. We're founded as a not-for-profit laboratory in 1997 to promote the use and acceptance of in vitro test methods for toxicology. And we work closely with industry and regulatory agencies, as well as provide a number of opportunities to educate the public. And IIVS has recently celebrated its 20th anniversary as a contract research organization. And we've also been able to celebrate our grand opening of an in vitro respiratory toxicology laboratory. This opening has been made possible through numerous contributions, and in particular, a very generous contribution from the People for Ethical Treatment of Animals to support the equipment needed for in vitro respiratory toxicology laboratory. So what does it mean to be a not-for-profit organization? Well, first, there are no owners or shareholders. We're supported by contract laboratory services, so we are a contract research organization, and through program contributions for education, outreach, and advocacy activities. The latter being through contributions typically from NGOs and some from industry. We have significant experience in optimizing, standardizing, and validating in vitro test methods, particularly in the GLP, Good Laboratory Practices environment. We also have success in bringing industry and regulators together to identify non-animal testing approaches that meet both of their needs, and we have several cases and examples of those activities. So being a not-for-profit really gives us this opportunity to be a neutral party when choosing the best in vitro test method, whether working for industry players or interacting with regulatory agencies or with animal welfare groups. So there's three prongs to our activities, and the first would be in the science. We conduct in vitro contract research testing on a daily basis, and we have participated in numerous validation studies over a 20-year history. We supplied in vitro testing services to hundreds of companies for typically thousands of products each year, as well as chemicals and ingredients. In this graphic, you can see a number of different activities in our laboratory in conducting these test methods. One of the things we learned from conducting these methods on a daily basis is that we learn how to handle a variety of different product types, different test materials, challenging materials, and understand which materials are best suited for different test methodologies. And that really helps us then identify the best test method for those products. We also have an education function. We teach the methods. We hold workshops, hands-on training activities, lecture courses, and training purposes for a number of organizations, as well as for international regulatory bodies and governing agencies. We have an annual workshop that we typically host for 4 days, which has both lectures and hands-on activities. Some of our workshops are simply one-day lectures, whereas others may be multiple-day workshops to really teach the fine details on conducting these test methods. And so in our mission, we seek to bring together this practical knowledge that we bring or obtain from the bench level working on a daily basis and seek to disseminate that through the education functions, through these training workshops. We're also advocates of the methods to numerous stakeholders. And with this, bring this together, we really hope to have an increased use and regulatory acceptance of these test methods by industry, as well as regulatory agencies overseeing their activities. Okay, so now I'll go into an overview of some of the non-animal test methods and really start with the simple concept that in vitro test methods were originally designed to examine fairly simple mechanistic activities in tissues to be able to relate to what happens in vivo. These methods were typically reductionist in nature, and so they really allowed us to focus on the individual tissues rather than looking at the whole animal. And in particular, this concept was applied to early on to addressing eye and skin irritation historically conducted in the Draize test in rabbits. And so the early thoughts were to reduce the number of animals used by applying in vitro test methods. The initial focus, as I mentioned, was on eye and skin toxicology, particularly for commercial personal care and cosmetics applications. By utilizing sound validated non-animal test methods, we really expect to replace these Draize tests for eye and skin irritation and corrosion completely. We can apply a number of different interesting test methods, from monolayer cell cultures to three-dimensional tissue constructs and ex vivo tissue models, all of which we will cover here shortly, with the ultimate goal of not using any animals for testing in the future. And in fact, as the technologies move forward, we're excited to see new applications such as the organ-on-a-chip concepts to really begin to move from the organ and tissue-specific predictions to perhaps a whole animal-type replacement concept. So as I mentioned, the in vitro test methods are typically reductionist in that one can really focus at various levels by using in vitro methodologies at those tissues or organs of interest. In this case, when we think about the exposure and consequences of chemical exposure to the eyes, we could either look at the whole globe exposure. Perhaps there's interest in looking at the lens and iris. Perhaps we were interested mostly in the epithelium, stroma, and endothelium of the cornea. We can even get more reductionist in looking at just the outer epithelium, or perhaps just this outer layer of cells covering the cornea. And each of these tissues have various consequences after chemical exposure, and it's important to be able to understand what those events are and be able to model those in vitro. For example, by looking at what's happening at the cornea level, we can look in utilizing ex vivo tissues such as those from the bovine cornea in the BCOP assay, bovine corneal opacity and permeability assay, or in the isolated chicken eye assay. These tissues are obtained as byproducts from abattoir activities. Perhaps more interestingly, for milder materials, we can look at what's happening at epithelium level, and there's a number of reconstructed tissue models now that focus on reconstructing the human cornea and the human corneal epithelium in three-dimensional constructs, and these are typically now used in both research settings as well as in regulatory classification labeling. And then lastly, if we just look at what's happening at the outer epithelium, there are a number of 2D, two-dimensional monolayer cell systems that could be utilized and applied for just these initial events that happen at the initial contact with the eye or the cornea. And so as part of this overview then for the remaining part of my introductory here, I will look at four major groups of non-animal test methods used in research and in regulatory safety testing of chemicals and products. And the first group would be the in vitro monolayer cell culture systems. These are the simplest systems, but a number of them have current applications using regulatory classified test methods in the process. And the first would be the 3T3 neutral red uptake phototoxicity assay, for which Alison Hilberer will give a detailed presentation. The second area is in the use of ex vivo tissues and organ systems. These include excised corneas from animal sources, and in particular, actually using ex vivo human tissue from human donors, and in particular, a regulatory test method that utilizes ex vivo human skin for predicting the permeation of chemicals through human skin. The third area will be in the in vitro reconstructed tissue models. So these latter two are much more complex than the simple monolayer systems and have a number of test methods that have now been accepted for regulatory purposes, including for skin corrosion and irritation, these of which Dr. Amelia Costin will give a presentation utilizing these models. And then the last category are non-cellular In Chemico test systems. And again, Amelia will cover the Crostex In Chemico type test system utilized for skin corrosion prediction. And Dr. Tinashe Rowana will give an overview of the In Chemico approach to looking at the reactivity of potential skin sensitizers to peptides. Before we move on, there's some practical considerations for testing. And the first thing to think about is, what is the testing for? What is the purpose for? Is this for product development, for product stewardship, or for regulatory classification and labeling? There are different test methods and approaches taken for each of these arenas. One also has to consider the availability of selected test methods, as there may be availability issues in regions, perhaps in the availability of some of the tissue models or tissues. There may also be some political barriers to obtaining certain test methods across government barriers or across country barriers. Perhaps the most important area for us to discuss here for the remaining presentations by my colleagues will to consider the following issues to select the most appropriate test methods. First, what is the physicochemical properties of the chemical or formulation? Is it a liquid? Is it a solid? What is the solubility, its viscosity, its pH? Some of the test methods are solution-based assays and may be incompatible with aqueous insoluble materials, so we need to have strategies for handling those type of chemicals or formulations. And most importantly, think about how to model the exposure conditions of each of the chemicals or formulations, as this has a significant impact upon the selection of the test method. And just as a point to make, in all in vitro test method testing, whether it's for regulatory purposes or for in-house, consider that one should include negative controls, positive controls, and have appropriate acceptance criteria. Concurrently tested benchmarks or reference materials are often very helpful to evaluate the results from these in vitro test method tests of individual unknown chemicals and help relate those back to what we know about the same behaviors of those chemicals with known benchmarks or reference materials. Okay, so we'll proceed now to the monolayer cell systems for product safety and regulatory testing. And I'd like to start out with some applications and limitations of each of these categories. So the first monolayer cell systems include those cells that we typically ramp up in flashes cells and seed into multi-well plate formats, or what has traditionally in the past been done using large format 2D type dishes. Typically, all the test applications occur within the multi-well formats. So you're applying test chemical to the cells. The cells are grown in 2D, and one then evaluates for some sort of endpoint response, whether it's for cytotoxicity or perhaps a gene expression change or some other type of evaluation. Essentially, all the activities are still happening in the 2D monolayer system. And so in this graphic here, the biologist here is reviewing what appear to be epithelial cells after treatment. These in vitro monolayer cell systems are generally very easy to conduct, and oftentimes we utilize cell lines, perhaps more appropriately primary cells, in the test method. They're generally quite rapid to execute. They're very well amenable to high throughput robotics. These methods are typically used for evaluating chemicals or ingredients rather than formulations, the latter being difficult and challenging sometimes with aqueous soluble systems, particularly if you have hydrophobic ingredients. These methods really allow us to look at mechanistic modes of action. This is really highlighted by the ToxCast program and the Tox21 approaches to being able to understand what type of changes happen at the cellular level after exposure to chemicals. Most of these methods are highly machine-scored endpoints rather than subjective endpoints, and they're typically geared towards potential hazard identification. Some sample applications of these 2D monolayer cell systems include evaluating chemical or active ingredient toxicity. There's some applications used to predict systems or organ toxicity. Typically under the Tox21 concept, they're designed to determine perturbations of normal pathways, early markers of changes in cells after exposure to chemicals. An application was validated for selecting starting doses for rodent oral LD50 studies, and there are a number of other approaches to be taken. And I think it's really a person's creativity can be utilized to apply each of these test methods for various purposes. But there are also regulatory assays, such as the screen for phototoxic potential, which is under the OECD Test Guideline 432. Other methods may include ocular irritation testing, such as that for the short time exposure assay and the cytosensor microphysiometer assay. I think the latter of which will be covered in the ocular irritation presentation. And one can use the 3T3 nucleotide uptake screen for acute oral toxicity. And lastly, Dr. Tanashi Ruano will cover the application of a keratinocyte assay, a reporter cell, that allows you to detect the potential for those chemicals that are typically chemically reactive and likely to be skin allergens. So these simple monolayer assays do have some limitations, and the first of which is aqueous insoluble materials are challenging to utilize in these test methods since the materials are diluted in the aqueous media. This is a particular issue that has to be evaluated before testing and is a concept that Alison Hilberer will cover in the application for the phototoxicity assay. There are effects of dilution, buffering, and dilution effects of the media which may mask the toxicity of the neat materials. And pharmacokinetics are poorly modeled as these simple monolayer models typically do not model barrier function of tissues. These test methods typically lack realistic cell-to-cell contact, and this may impact the cellular responses. In fact, these cells cultured on 2D have potential impacts of being cultured in 2D that de-differentiate their performance and may be notably different from their normal behaviors in vivo. And this is illustrated by this graphic here that in the toxicology using human epidermal keratinocytes for evaluating skin irritation, an application that was utilized 15 years ago as a first concept, we can see that these cultures here, these 2D cultures here, have very little resemblance and show no barrier function relative to the cross-section of human skin here. And so we really have to ask the question, do these cells here really resemble the epidermal cells in three-dimensional in vivo setting? So what do we do? Then we perhaps consider more complex models, and some of the early models to be viewed were ex vivo tissues and organs for product and regulatory safety testing. Now these ex vivo models allow testing without the use of live animals. The excised tissues directly correlate to the tissues of interest, and in particular in replacing the Dray's test, the isolated rabbit eye test was the first application of using ex vivo tissues to mimic what was happening in the Dray's test. And similarly, bovine and chicken corneas were utilized to do the same and to build test methods that utilized tissues from animals that were already harvested for abattoir purposes. And human skin, as well as other human tissues, have rapidly been integrated into in vitro or ex vivo test systems, and in particular, human skin is utilized to routinely evaluate the potential for permeation of chemicals across human skin. The ex vivo tissue or organ systems allow us to expose chemicals very much in the same fashion as occurs in vivo. So there are no dilution effects from making dilutions in aqueous media. You can apply the formulation or material exactly as it's exposed in vivo. They tend to be cost effective and generally available, although as I mentioned, in some arenas, some excised tissues may not be readily available, either for political or for geographical reasons. The models also have very relevant mechanisms of action as well as some side actions, and the endpoints may also be readily machine scored, just as we can machine score 2D test systems. But as any assay has, we also have to consider the limitations of ex vivo assay systems as well. And the first may be the issue of availability. Human tissues of exceptional quality are often difficult to obtain, so then sometimes we have to look at other sources of ex vivo tissues. There may be also variability issues, as the tissues may differ from trial to trial, particularly from human subjects. If the tissue is non-human, do we question the relevance? We should. We should definitely consider that. And one must be careful to not over-interpret the results. And I think this is really important as we look at, in particular, some of the models that mimic the ocular exposure. We forget the fact that these models have been excised, have been removed from the whole animal experience, and perhaps the endpoints we're querying are still not covering all the endpoints that we should be looking at. Excised tissues no longer have the support of whole body accessory functions, and so we don't have the potential for full metabolism as if the tissue is available in the whole body experience, or that of inflammatory responses. And so we have to consider how this might impact our toxicity predictions. The third arena has really looked at taking the concept of 2D cell culture and beginning to utilize technologies to make three-dimensional tissues with differentiated tissue states. And this really comes from the technology utilized for creating tissues for transplant purposes. Also, similar to the ex vivo tissues, reconstructed tissues better model tissues of interest relative to monolayer systems. We can expose the test substances just as in vivo. So again, we can do a topical application for skin or eye models or for mucosal tissue models. Unlike the ex vivo tissues, standardized manufacturing ensures standard platforms, and so we can expect a high level of reproducibility owing to good manufacturing practices by the suppliers of these tissue models. And again, we have very relevant mechanisms of action, and in particular with human tissue cells or tissue models replicating the human in vivo setting, we may particularly have very specific human lock and key mechanisms of action available. And again, the endpoints may be machine scored. And again, unfortunately, we always have to consider some of the limitations of all tissue systems, or all assay systems in particular, and the same holds true for reconstructed tissues. Tissues should be readily available, but we may have some challenges in importing some of these tissue models into various countries. Tissue models tend to be costly. They're quite complex tissue systems. We also rely on a relatively small number of manufacturers worldwide for good quality commercial tissue models that are cultured under good manufacturing practices. And in fact, even if some of the tissue models are useful for a variety of regulatory test methods, even among the tissue manufacturers, tissues differ slightly. They may have some common performance attributes, but there are differences among each of the tissue models, and we need to understand that when we apply one model and consider replacing one model with another manufacturer's analog. And lastly, still relatively simple, these models do not have support of whole body accessory functions, and in fact, they may not have all the cell types or all the cell functions of an ex vivo tissue. Particularly when we think about the ex vivo cornea relative to the reconstructed corneal tissue reconstruction, we have analogous tissues in the three-dimensional reconstruction, but we don't have quite the complexity of the ex vivo tissues. So again, we have to ask the question, how might these changes impact our toxicity predictions? And just as with the ex vivo tissues, we have to be very careful not to overinterpret. It is very easy to do so. Our last area for introduction here is in the use of non-cellular or in chemico test systems for product safety and regulatory toxicology testing. And these systems, again, are quite different in the fact that they do not require cell culture facilities or cell culture expertise. They rely more on the chemistry for their endpoints. They may be relatively inexpensive to conduct, and even the reagents may be relatively inexpensive, as some of the test systems, non-cellular test systems, were proposed early on as replacements to the Dray's test. Some of these test methods allow exposures as in vivo and may have a topical application approach. And finally, the standardized manufacturing or processes used to prepare these tissue models, or these non-cellular models, are very reproducible. Okay, just as we noticed with the other test methods, there are limitations of non-cellular or in chemical models. Some process-based test methods require specialized equipment. This is the case for the direct peptide reactivity assay, which relies on the need for HPLC analytical tools, equipment, and expertise. There's also reliance on a limited number of manufacturers of these commercial platforms. In the case of the Crostex, there's a single manufacturer of that test system. However, the test guidelines have been written to allow for innovative applications of similar or Me-Too type concepts so that other manufacturers can make very similar type platforms as well for regulatory testing. They do lack the complex biological responses, so we really think about these models as being about as reductionist and focus on a specific event or perhaps even a key initiating event as possible. For example, these models are unlikely to address metabolism unless one includes that type of component in the test design. And they certainly lack the inflammatory mechanisms that need to be modeled and that occur in vivo. And so we have to ask a few questions. What are the artifacts that may color the relevance? For example, false positives that occur in the direct peptide reactivity assay from non-binding peptide oxidation need to be addressed, and one needs to have a study design that includes metabolism and/or preemptive oxidative activity on the test chemicals. And so as we close on this concept here, here's some thoughts to summarize these discussions as well as some thoughts to move onward. Each of the assay systems have useful applications, but they also have limitations. First, in the relevance. Are the test system and the cell type and exposures and endpoints appropriate? Are they relevant to the prediction you're trying to make? The exposure is quite important. The application of chemicals, substances, or formulations to the tissues need to model the in vivo exposures. And so if they differ, we need to think about the appropriate test approach to meet the exposure modeling goals. And the gaps. Which models provide all of the required information? Which are only supportive? When we think about the application of the three-dimensional tissue constructs and the ex vivo tissues provide the vast majority of the information we need, but there are still gaps, and we need to be able to address those as well. Certainly, this then concludes to the fact that one size assay does not fit all. We need to apply a tiered approach, complementary testing approaches, and assessment strategies so that we can address each of these issues of relevance and gaps in and limitations of each of the test methods so that we can build a better picture for the in vitro test outcome. So I thank you, and we'll move on to the next presenter here shortly.